Hello, Mount Aloysius College students, faculty, and community members. My name is Benjamin Melly. I'm currently a student at Mount Aloysius College. I come to you with a question. Have you ever considered how soap affects the growth of bacteria? The discovery of soap dates about 6,000 years ago, around 2800 BC. Ancient Babylonian excavations uncovered cylinders with inscriptions for making soap. In 1500 BC, records from ancient Egypt described how animal and vegetable oils were combined with alkaline salts to make soap. Soap ingredients may have changed into ancient Babylonian times, but what does soap do when you use it? Soap is a surfactant due to its molecular structure, which is comprised of two distinct ends, a hydrocarbon end, which is lipophilic and nonpolar, while the second end is hydrophilic and is polar. The nonpolar end is capable of dissolving nonpolar molecules, while the polar end is capable of dissolving polar molecules. The efficiency of soap is affected by various factors such as the pH, the composition of the solvent, which is what you're washing away, and the temperature. The nonpolar tail is capable of dissolving nonpolar molecules such as oils and grease, and the polar head is capable of dissolving polar molecules such as water. For the past few months, I've been trying to determine what soap brand is more efficient in inhibiting bacterial growth. I selected 4 different brands of soap to study: Ivory, Irish Spring, Dial, and Dove. Each brand was selected for its main antibacterial component. In Ivory, sodium toluene is the main antibacterial component. Sodium toluene is a surfactant, which helps water remove oil and dirt from the skin. Sodium hydroxide is found in Irish Spring. Trichlorocoraban is found in Dial soap. Sodium lauryl isethionate is found in Dove soap. The detergent molecules of soap encase the soil. Then the soap is pulled away from the surface it was attached to. Surfactants are used in cleaning and can kill bacteria by interfering and breaking up the cell membrane components such as lipids and proteins. The hydrophobic surfactant tail embeds itself in the lipid layer surrounding the cells and causes it to break apart, which can easily be washed away with water. An inoculation introduces live microorganisms into a medium where they will grow and reproduce. In this experiment, Staph aureus and E. coli were introduced to a nutrient broth. Above is shown how the inoculation was performed. The shaking incubator maintains optimal conditions for incubating microbes. This equipment is used to evenly distribute the necessary oxygen and nutrients for the microbes to reproduce. In this experiment, each tube with separated samples of Staph aureus and E. coli were in the shaking incubator for 24 hours at 200 RPM. A serial dilution is a series of sequential dilutions used to reduce a dense culture of cells to a more usable concentration in order to count the colonies of cells plated on the agar plate. Typically, there should be 30 to 300 cell colonies. The dilution estimates the number of cells plated. In this experiment, there are 5 sequential dilutions. In this representation, the test tubes were dyed in order to show the dilution of the bacterial culture. Plating cells is used to separate microorganisms contained within a small sample volume, which is spread over the surface of an agar plate, resulting in the formation of discrete colonies distributed evenly across the agar plate. Agar is a nutrient-rich medium that promotes cell growth. In this experiment, 100 microliters of E. coli and Staph aureus were plated on 12 separate plates. There are 4 plates for each brand of soap and 2 plates for a positive and negative control. The glass rod sanitized in this video is a cell spreader. The cell spreader ensures an evenly distributed lawn of bacteria on the agar plate. The disc diffusion test was used to determine the susceptibility of bacteria to the different brands of soap, and an effective inhibitor will produce a large zone of inhibition, which means the saturated disc inhibited bacterial growth, while an ineffective inhibitor may not affect bacterial growth at all. For the purpose of this video, I used my own hand to sud the soap. In this experiment, a fellow researcher sudded the soap. The disc used in the disc diffusion was saturated in the suds of each soap. For the positive control, an antibiotic, gataflaxin 5, was used, and the negative control was nutrient broth. After each disc was plated, the plates were then placed in the incubator for 24 hours to promote bacterial growth. This graph depicts the zones of inhibition measured in millimeters on the y-axis and the soap brands on the x-axis. Ivory was the most effective inhibitor with an average zone of inhibition of 14.5 millimeters. Dove was the second most effective with an average zone of 11.3 millimeters. Irish was the third most effective with an average zone of 7 millimeters. Finally, Dial was the least effective soap with an average zone of 5.8 millimeters. The positive control, gataflaxin 5, an antibiotic, had an extremely high zone in relation to the soap, as well as the negative control. The negative control, nutrient broth, promoted bacterial growth instead of inhibiting it, and rarely had any zone. This is an example of the trial 3 against Staph aureus for ivory soap. This zone of inhibition was 7 millimeters. This is an example of how effective gataflaxin 5, the antibiotic, was in inhibiting bacterial growth. This plate is an example of the negative control in trial 3 with the E. coli bacteria on the plate. As you can see, there was no zone of inhibition. Further research on this topic could test the effectiveness of the soaps Ivory, Dove, Irish Springs, and Dial in hand washing. Thank you for your time today, and I hope you were able to learn a little bit about the effectiveness of hand soaps in the inhibition of growth in bacteria.